We're going to change gear a little bit, and I'm going to talk about fungal pathogens, so completely different kingdom. So most of you probably, when you think about fungi, you think about some of these things, right? Bread and yeast, maybe blue cheese, which is made with fungus. Corn, it's actually mycoprotein. They also make a lot of the medicines that we currently use. Penicillin, one of the great examples, but also the statins. But they can also be quite detrimental. So this is sort of on a local scale. If you leave out fruit for a bit too long, you get some fungal growth on it. But on a larger scale, we've seen some outbreaks, things like the potato famine in Ireland, wiped out complete potato population. And also currently the banana outbreak, so that's a fusarium disease, very resistant to anything we've got and very detrimental to plantations, mostly in South America. But when we think about sort of fungal diseases that we actually work on, they go from sort of the topical ones, and I've saved you a picture of a fungal nail and skin infection, but also things like mucosal candidemia, so this is that sort of plaque that you see on the tongue, to really severe invasive diseases, things like invasive aspergillosis, so this is one of our patients, their lung. Hopefully you can appreciate that sort of that white mass that you see there is all fungal hyphae that are growing within that lung. The only thing that we can do is a year-long antifungal treatment, surgical resection of the whole lobe, so really severe infections. But it even goes as far as a brain infection, so this is cerebral cryptococcosis. Again, I hope you can appreciate that's not a normal brain. There's a massive lesion there, which is all cryptococcus fungal. Now, fungi, most of the infections are actually derived from the environment. That's why sort of this One Health approach is really important, and that's what we take in the lab. So we acquire fungi via aerosolation, so spores are in the air, we breathe them in, they can cause infections in the lung. They live on animals as commensals, so even on your pets, if you sort of pet your cat or your dog, you're probably transferring fungi. And they live on plants and soils, so in agriculture we see a lot of farmers with sort of infections as well. Now there's always the thing, it can cause an infection in the right patient via the right exposure route. So there's lots of different exposure routes that you can take, lots of different settings, lots of different patient populations. But generally the patients that we see with fungal infections are immunocompromised. And sort of the numbers that we see, these are estimates. We're not really clear about what the actual fungal burden is. This is sort of one of the best estimates we've got. But we're talking about millions of infections annually. A lot of them are chronic infections, but we also see things that are sort of more acute, like invasive aspergillosis, mucormycosis, and also things that are relatively quite common, things as vulvovaginal thrush, and things like skin, hair, and nail fungal infections. But this is sort of the landscape that we're currently working with. And as the title of my talk said, the landscape is ever-changing. This is where the climate change comes in. We see sort of slow increased temperatures, more extreme events, natural disasters, but even things as far as sort of seeing a change in complete biosystems and ecospheres that also change what fungi grow in certain environments. Now that's not the only thing that we look at. We also consider changing environments, things like chemical input. And what we put in the environment is actually millions and millions of tons of fungicides annually. That's mostly in agriculture, so fungal diseases in agriculture, as I showed you at the start with the potato and the banana, are in agriculture, plants are really at risk of fungal diseases. So we spray the land with incredible amounts of fungicides. They obviously end up in the environment, and that's also where our pathogens live, and they are exposed to those fungicides and become resistant. Actually, really interestingly, most of the antifungals that we use in the clinic sort of have the same mode of action as the fungicides that we use in agriculture. So you can already see that there's sort of that problem of dual use and resistance there. So in our lab, we basically, a cornerstone of our lab is how different environments give rise to changes in fungal infections. That goes from a global scale all the way down to sort of the real microscopic level. And today I'm going to talk mostly about one of our favorite fungal pathogens, which is Aspergillus fumigatus. It can grow sort of like what you see here on the left. That's on a plate. It grows these tiny little colonies, nice and green. This is probably one of the ones you see at home as well. It likes to grow on fruit. And it forms millions and millions of spores, and they are really easy to aerosolize. Basically, if you get a little bit of wind on one of those plates, they go everywhere. And that also is what happens in the environment. It also likes to live in agricultural fields, again, where tons and tons of these fungicides are sprayed. So that sort of resistance in Aspergillus fumigatus to fungicides, and therefore antifungals, is an environmental problem that we're currently trying to deal with. This is one of the earlier studies that we did. We actually looked in the UK and the British Isles, and we collected Aspergillus fumigatus from the environment, about 250 different isolates from all over the country. And we whole genome sequenced them. And we actually looked for resistance markers to one particular class of fungicides, which is the azoles. The azoles are also the first line therapy in the clinic, so it's sort of really the cornerstone of treating fungal infections. Now, what's really nice from a genomics point of view is that environmentally acquired resistance has a real genetic hallmark. While normally, if it's a chronic infection and in the clinic, you see sort of point mutations that are listed on the right here in sort of the green colors. If it becomes resistant in the environment, it has a tandem repeat in the promoter of the drug target and one particular point mutation. There's multiple combinations, but there's always a tandem repeat and there's always a point mutation. So we can do some really interesting analysis then. And what we found when we whole genome sequenced about 250 isolates, we found that the cross-resistance to clinical and agricultural antifungal classes really has a population structure. So this sort of clade A that we saw here on the right has most of the resistance, environmentally derived resistance. So there is something that is driving the acquisition of resistance, because this is based on whole genome sequencing, and we were wondering what that actually was. So this is that same tree again, just represented slightly different. Anything that's resistant has the outer ring in orange, but what we were interested also in is actually DNA mismatch repair. There's a lot of data out there already from bacterial pathogens, but also some fungal pathogens, that if you've got mutations in any of these genes, you mutate much more rapidly and therefore can become resistant much more rapidly. And I hope you can see here that almost all the strains that had this TR34 L98H mutation, aka azole resistance, also had a mutation in this gene called MSH6. Now that's one of those DNA mismatch repair mutants. And when we look at how fast it can acquire resistance to a different kind of drug, if we just take strains that don't have that mutation, so here in blue and in black are lab strain, there's sort of low level of mutations to acquire resistance to a novel drug. But all these strains that have the mutation actually acquire about 6 times more rapidly resistance to a new drug. So we actually think that that is driving resistance more rapidly in that clade, and actually that's why we see that population structure. Now, interestingly, it's only 6-fold. If you knock out the gene, it's about 120-fold. So we think there's an actual balance between mutating more rapidly and sort of a detrimental effect where you mutate so rapidly that you just die off because of deleterious mutations. So that's something that we're currently following up as well. Now, we wanted to do this on a larger scale, so we were funded, luckily, to do this as a global study. And we collected about 1,500 different isolates from all over the world. I hope you can kind of see where they're all from, with a lot of collaborators, and we whole genome sequenced them all. And we actually wanted to see what was driving resistance across the world. And what we could do is we can actually monitor things in time, because we will know that when they were collected and where from. This is sort of all the grey dots that are still susceptible. They're sort of from the 1900s, 1920s, 1930s, 1940s, 1950s. And then actually in sort of the 1980s, we see this first resistance, this TR34 pop up. All again in that sort of similar clade that we saw before. Then sort of in the 2000s, we see really rapid acquisition and really rapid expansion of all these strains. We see a novel mode of resistance in red, and actually sort of in 2010, this thing really starts to explode. Now, what we can do as well there is we can actually map that back to where things come from. With fungi, that's a little bit more tricky because they recombine and, you know, they love to merge and fuse and all that. So we actually had to do this via the mitochondrial genome, which is nice and clean. But then you can track sort of where things come from. And there was a real hotspot of resistance, sort of actually right here in the UK and sort of in the Netherlands. That was sort of within our spot, which was great for me because I'm originally from the Netherlands and I work in the UK, so it's very close to home for me. But that was actually what was already thought, that that's where resistance comes from, because in the Netherlands we've got this this sort of thing that we do for our tulip bulbs is when they're harvested, we dip them in a bath of azoles and then we ship them off across the world. So actually we're probably spreading a load of azole resistance across the world that way. Obviously the government and the tulip growers were very happy to hear that, but we're still trying to figure out how to work this into policy. But that's sort of looking back at azole resistance, which, you know, has been known since the 1980s. What we also want to do is look forward. So we generated a spatial model where we could actually see where in the soil does Aspergillus fumigatus grow. You get a sort of a probability map. So anything here in red is where it's very likely that Aspergillus fumigatus can grow. Anything in blue is where it can't grow. It kind of made sense with all the environmental data that's out there. What you can then do is look forward. You can project climate, different climate outcomes onto that. We did a couple. One where actually it gets really hot because we keep burning fossil fuels, and one where all the interventions are in use, so it actually cools down a little bit. And oh, that's very well. You just have to trust me on that. So I'm going to skip right over this. When you actually do that in time, you can see Aspergillus fumigatus sort of goes more northwards, and other species that are more tropical actually move sort of more polewards. Hopefully that one works. Yeah. So we made a little graph out of that. So this is a different one, Aspergillus flavus, and we can really see sort of expansion into Europe, and actually it becomes less common in sub-Saharan Africa because it probably gets too hot and too dry there. So what that means is we can already start to prepare for what comes next in the next 50 years instead of looking back. And this is what the newspaper thought of it. If you've seen The Last of Us, then yeah, horrible things. We're also looking at novel drugs. So I was talking about how we've got different drugs coming through the pipeline. We've got a novel antifungal coming through the pipeline, olorofim, but at the same time, the same target is being used for agricultural fungicides. And we did a lot of mapping, so this is an in silico model where you see this new drug, olorofim, and this agricultural fungicide, iflufenaquin. They basically bind the same pocket in the same molecule. We did the experiment in the lab where you evolve a load of different strains to become iflufenaquin resistant, and actually, when you become resistant to iflufenaquin, you also become resistant to olorofim. So this dual use will still continue to be a future problem. Now the WHO kindly wrote about it. They actually want to do something about it. The UK also want to do something about it. The House of Lords and the House of Commons actually had a debate about it, which has been very useful, and you know, hopefully we can sort of make sure that this dual use problem is a problem of the past. Now that's not all that we do. We do a lot of different fungal pathogens. I just wanted to highlight, we've found some fungal pathogens in Antarctica. We do outbreaks in Brazil, different fungal species, and this is my PhD student who's trying to treat fungal diseases in the global south. So a lot of things going on. If you're interested in any of this, please come and chat to me in one of the breaks. So, in summary, fungal diseases are really under-researched, under-diagnosed, under-treated, but they really contribute to mortality. We need to understand the eco-evo disease pathways. Where does it come from? What does it do? How does it cause disease? And that's why we use predictive models and experimentation to safeguard the future of antifungals and our patients. And that leaves me to thank an awful lot of people. All this work was done in Manchester, but hopefully we'll get to do some more here in Oxford as well. And with that, thank you very much. Happy to take questions. We're going to